going to talk about T cell development. Couple of background or sort of administrative pieces of info. Um, all of the information in terms of a checklist, rubric, all sorts samples, all sorts of other stuff, all is posted on Moodle now as for the lab report. Lab report's still, you got time. There's a draft that's due, I think, November 7th. But all of the stuff for it is due, and in fact, you made some graphs yesterday to go into it. So like, you already started. Good job. We are not going to have lab next week. So next week is the week that I give you off because I made you have lab the first week. So that's, I wanted to make sure you had all that information if you wanted to use that time on the lab report, but you can use it however you'd like. I will also tell you I'm going to be away some of next week. Wednesday's class is going to be on Zoom next week, but I will be here on Monday and Friday. I might be a zombie on Friday, but I will be here. The answer key for exam 1 is posted. That's actually like typed and like nice and legible, as opposed to the unlegible version that I used when I was grading. I will also tell you that when I was going through I was typing one part of it, there was one question where I was like, that's weird. I feel like I graded this question. I don't like how I graded this question. And so I went back and gave everyone an extra point. Because I'm pretty sure I took off a point for everyone on something that now I was like, that was wrong. I shouldn't have taken that point off. So if you now look at your grade on Moodle, your grade is 1 point higher. All of that key is set. Also remember the antigen presentation problem set is due today by 5. So please make sure to send that to me if you have not already done so. So like I said, today we're going to think about T cell development. And T cell development occurs in the thymus. So basically, we're going to be seeing a process that is really similar to the process of B cell development that we previously saw. But one of the big differences is that our precursor cell has to migrate to the thymus in order to do T cell development. And as we talk about T cell development in the thymus, I will reference different areas of the thymus, specifically the cortex and the medulla, which you can see here. The cortex is the darker colored area. It is much more densely packed with cells. So we'll see certain events happening in the cortex, other events happening in the medulla, which you can see is more lightly colored, and that's really because it's less densely packed with cells. And in both cases, we have a lot of structural cells of the thymus. They're in pink, and they're in white, and orange, and yellow, all sorts of them. Those are known as the thymic stroma. stroma, or stromal cells. That's stroma with an L at the end. And you can see the developing T cells, which are in blue in this cartoon, are interacting with those stromal cells a lot to help their development. And that will be pretty key throughout this process. So we've got to go through the steps of T cell development. And to think about T cell development, I'm going to start by making a little table over here about B cell development so that we can do a little compare and contrast. So what I want us to remember is we had pro B cells. I'll look at this first. All right, so here's my overview of B cell development. We had pro B cells, pre B cells, and immature B cells. The pro B cells had two major events they were doing. First, D of the heavy chain rearranged with J of the heavy chain, then V of the heavy chain rearranged with DJ. And there was nothing on the surface of those pro B cells. Then we had the pre B cells, which had the pre-TCR on the surface, the heavy chain they had made previously, along with a surrogate light chain. In the case of B cells, the surrogate light chain's names was two proteins, and the names were V pre-B and lambda 5. And then the pre-B cell did a bunch of actions, including things like allelic exclusion of the heavy chain, surviving and proliferating, and then finally doing light chain rearrangement. And if it was successful there, then it was an immature B cell, which did a bunch of central tolerance testing and had the BCR on the surface. So this is our B cell development overview. So I want to give you a brief overview of T cell development now, using this table. There you go. So if you notice, in making at least a very broad overview of T cell development, I just took this table and I erased the B's where it said B cell, and I wrote a T for T cell. And so at some level, you can see that it's really similar to everything that we've talked about with B cell development. The place where this gets a little bit different is that we don't frequently use the terms pro T cell, pre T cell, immature T cell. You could, but we actually have some other terms we use instead more often. And a lot of the things that are useful about those terms is they actually will make these two steps separate steps, or they'll make these sorts of steps separate steps. So we're actually going to have a name for this cell that's doing this kind of rearrangement versus the name for a cell that's doing this kind of rearrangement. But in the big picture, the overall scheme of what's going on in T cell development is the same scheme that you've seen before. When we look at the thymus, we tend to distinguish the developing cells with a few different markers by flow cytometry. When you take the thymus out of an organism, you generally can get mostly T cells, which you in fact saw with your mouse. And so you can kind of assume everything in the thymus is T cells. Yes, there are all those structural cells, but when we look at this kind of a plot, we'll just say that we're only looking at T cells because we pretty much are only looking at T cells in this situation. And we find that those developing T cells in the thymus can fall into 4 different populations based on flow cytometry staining. And when we're doing this flow cytometry staining, we're using either CD8, and CD4. So we'll have CD8 on one axis, CD4 on the other axis. And the 4 populations that we see in the thymus are this population that are negative for both CD4 and CD8. They're usually referred to in this case as the double negatives. We have CD8 positive, CD4 minus, which are single positives. We have CD4 plus, CD8 minus, which are the CD4 single positives. And we have CD4 plus, CD8 plus, which are known as the double positives. One of the things that's really key is you can actually see the sort of shape of this sort of shape is very characteristic. This is, if you look at a thymus and do a plot of CD4 versus CD8, this is what you should always see. If you're going to draw it, one of the things that I want you to notice is that if you look at your, look at the double negative population, the double negative population is the smallest. It's here only about 3%. So it's a little tiny circle. You can see that the CD8 population is a bit bigger. CD4 is even a bit bigger than that. But double positive population is huge. And so if you're drawing this, I'm particularly looking for your double positive population to be the biggest by far. You can see here it's shown as being about 80%. I'm looking for the double negatives to be the smallest, and I'm looking for the other two to be in between, though I'm not going to like measure them with millimeter rulers to figure out which one's bigger. I'm just going to say those both should be in between. Double neg should be the smallest, double positive should be the largest. And these double positive cells that express both CD4 and CD8 are a unique population of developing T cells that are only found in the thymus. So you should never otherwise, we're outside the thymus, you never find a cell that has both CD4 and CD8. That's only something that happens during a specific stage of T cell development. And so we often are going to talk about things like our double negatives and our double positives or our single positives when we think about T cell development. We also have realized that these four populations of cells are heterogeneous. So all the cells in each of these four populations are not necessarily all the same. And specifically, we can take the double negative cells and further divide them up. So we can gate on the double negative cells and divide them up further. I'm showing you a version of this from a paper here. Yes, in this paper, they probably had way more cells than they did here to get that many dots of double negatives. But what you can see is that we can, again, do a flow cytometry plot. Now we're going to use a marker called CD44 on our y-axis and a marker called CD25 on our x-axis. And we're going to, again, get four populations. Since they're all coming from the double negative population, they all have DN or double negative in their name, and then it's 1, 2, 3, 4. So you can see we've got DN1, which is CD44 plus, CD25 minus. DN2 is CD44 plus, CD25 plus. DN3, CD25 plus, CD44 plus. DN4, CD25 minus, CD44 minus. And so what we now know, and again, you'll see much more of this as I go through all of this, is that when T cells come into the thymus, they come in sort of as double negatives. And in fact, they come in as double negative as DN1s. And then they become DN2s. And then they become DN3s. And then they become DN4s. And now they've finished their life as double negatives. So after their DN4s, they become double positive. They follow this little arrow. And then they're going to follow these other arrows later. You are going to see that in more detail on subsequent slides. So if we try to think about this in terms of the stages that we've seen previously, we can first kind of think about the pro-T cell. Pro-T cells are found in the DN1 and DN2 populations. So DN1 cells and DN2 cells would correspond to what we have on my little table to the whatever side that is, left or right, your left, my right, as pro-T cells. Yep, Brenna.
Just to clarify, are the main populations in DN3 and DN4? Are the main populations?
You know,
like how would we draw it if we wanted?
Yeah, I would say that DN3 and DN4 are bigger. I am not sure I've ever actually asked people to draw it. So, but yeah, DN3 and DN4 are bigger populations. Yeah, Shelby.
Are DN cells named that way because of where they develop?
Yes. So are named T cells because they develop in the thymus, but B cells are actually not named after bone marrow. But it would make all of our lives so much easier if they are. So, but right, it would help you. If you want, so in reality, B cells were named after this weird organ where they develop in chickens, which also starts with B. But if you want to remember its bone marrow, you are more than welcome to do that. If you don't want to think about chicken anatomy. So the DN1 population is actually the population that does the migration to the thymus. So it's the population that comes into the thymus. It's sort of that original cell that's like, I have decided to live the T cell life. The DN2 population is the population that's actually doing the heavy chain rearrangements that we see listed here. So DN1 is the cell that actually comes into the thymus. DN2 is the cell that is going to start doing its heavy chain rearrangement. One thing that you can notice here, again, this is something I'll say more about later, is that this textbook does mention that, in fact, the DN2 is trying to do rearrangement of delta and beta, both kinds of heavy chains at the same time. And it's actually also doing gamma at that same time. So I'm right now just going to say heavy chain, and usually that means beta. That's the one we talk about the most often. But in reality, as you'll see later, it is actually beta, delta, and gamma that are all rearranging in DN2. What? Chaos. And these cells are come in, so they enter the thymus right at the border between the cortex and the medulla. So you can see the DN1 cell comes in right at this border between the cortex and the medulla. And then the DN2 cell moves into the cortex and is spending time in the cortex. What you can also see here is that, cells that are this early, DN1s and DN2s, can still make some choices to go different routes of development if they want to. They're not 100% totally committed. They'll get totally committed later. Our cell has not yet decided if it's going to be an alpha beta T cell or a gamma delta T cell. So it's still kind of making some choices. It has not yet declared its major. Once our cell finishes being a DN2, it will have made a heavy chain. And once it's made a heavy chain, it will want to put that heavy chain on the surface of the cell in order to see if it's a functional protein and if it can get a signal. And as a result, it will be a pre-T cell because it's now putting its heavy chain, its pre-T cell receptor on the surface. And it's now, that cell is now a DN3. And so our DN3s are where the cells start being the pre-T cells. Just like in B cells, we need to have a surrogate light chain binding to the heavy chain as a part of the pre-T cell receptor because we have not yet made a light chain. So we can't pair the heavy chain with the light chain because we didn't make a light chain yet. So we've got to have some kind of surrogate light chain. The surrogate light chain is shown here and is listed here as PT alpha. In some cases, you also see it listed as pre-T alpha. So if you call it either PT alpha or pre-T alpha, I don't care. But you can see that it is pairing with the beta chain to allow for a whole T cell receptor to be signaling. It's also pairing with all of the CD3 chains at this point, so we can get a signal. One thing to also notice here is that this cell is a DN3, double negative. That means it doesn't have CD4 or CD8 yet. DN means no CD4 and no CD8. So we don't have CD4 or CD8 to pair with this yet because we haven't made them. But we do have the CD3 chains. Just like with the surrogate light chain in B cells, or with the pre-TCR, which contains a surrogate light chain in B cells, there's a little bit of a question about the T cell pre-TCR. Specifically, the question is, okay, if we need a heavy and light chain together to get a signal, and we don't have a light chain yet, then how the heck do we get a signal? We also don't even have a CD4 or a CD8 yet. So we want to test this heavy chain to see if it works, but we can't really get a signal very well. And what we think happens is actually that pre-T alpha can bind to other beta chains and sort of pull together multiple adjacent pre-TCRs to allow for signaling. And so it seems as though we can actually get a signal if we pull together multiple pre-TCRs, which is only possible with a functional heavy chain protein, because pre-T alpha or PT alpha can bind to the beta chain in both sort of both in sort of the adjacent beta chain, but also it can bind to other beta chains at the antigen binding surface. Once our cell is has made that heavy chain, it's going to do its selection for a good heavy chain, and it's going to do all of the same actions that we saw with a pre-T cell, with a pre-B cell. So first, we're going to see allelic exclusion of further heavy chain rearrangement. So the first thing we have to do is we shut off RAG. We say no more RAG. We stop making more heavy chains because we have a good heavy chain. Now that RAG is off, the cell can proliferate because we aren't risking DNA damage during the cell cycle. We also give that cell a survival signal, getting a little pat on the head that says, you did a good job, you're allowed to live. And those are going to be sort of things that are going to be happening with our DN3 to DN4 cell. As the cell continues, and so you can kind of see that here, that we're going to start this testing process in DN3. So we're going to start making the pre-TCR. We're going to start testing it in DN3. And we're going to do things like proliferation and allelic exclusion in DN4. So those are going to be kind of big events that are happening in those double negative cells. All of these processes, all of these processes are happening in the cortex. So our cell is going to move through the cortex and be doing these rearrangements of the T cell receptor. You can also see that our cell is going to be potentially choosing to become a gamma delta cell as opposed to an alpha beta cell during this time. And so we have to think a little bit about how that choice works, because it's happening in this sort of DN2 to DN3 time period. As I told you before, in the early stages in our pro-T cell, our T cell is actually going to start doing rearrangement of beta and delta, which are both heavy chains, as well as gamma, this light chain, all at the same time. So all three of those happen during the time that the cell is a pro-B cell or pro-T cell. You can see that here. And eventually, we're going to have allelic exclusion. Basically, we're going to say, no, RAG, you are off now. We're going to turn off RAG because the cell has received some kind of signal through a receptor on its surface. There are two possible options for what that receptor that's on the surface could be that gives the cell the signal to turn off RAG. Before it gets the signal to turn off RAG, it's chaos of trying to rearrange all three of these things. And so what you can kind of imagine is that it's a race between these two different kinds of receptors. Which rearrangement happens successfully first? Whichever one happens successfully first wins because it can give the allelic exclusion signal and stop the other one. So one thing that could happen is we could get a successful beta chain. And if we get a successful beta chain, then that successful beta chain can pair with the pre-T cell receptor, and that will inhibit gamma and delta from further rearrangements. Alternatively, the cell could make gamma and delta correctly. So this might be a little bit trickier because it's got to get two correct rearrangements, a correct gamma and a correct delta. Put them both on the cell surface. And if that happens, that signal turns off beta rearrangement. And so it's a race. Do you get beta made correctly first, or do you get gamma and delta made correctly first? Whichever of these things happens amongst the chaos gives you allelic exclusion and blocks rearrangement to go down the other path. And so here you can see the cell is going to be doing beta, gamma, and delta rearrangements. If it succeeds with gamma and delta, then it's going to do gamma delta life. If it succeeds with beta, then it's going to start going down the alpha beta track. If it actually starts going down that alpha beta track, it still has a little bit of an out to switch. But it's usually getting pretty committed. Though again, it does have a little bit of an out. And so you can see sort of this, again, it's happening right here, kind of at that DN3 stage where we're either going to make a beta chain or a gamma delta chain. If we make a gamma delta chain, we get a sort of DN3, and then the cell just becomes a gamma delta and is no longer part of our T cell development as we're talking about it. If the cell makes a beta chain, then it's going to keep going towards being a DN4, a double positive, eventually a single positive. It's going to keep doing all the parts of T cell development that we're talking about. And these two types of mature T cells that eventually end up in the periphery, either the gamma deltas or the alpha betas, will have different jobs. And they have different jobs probably because way early when they're DN3s, they got a different kind of signal from a different receptor. And so that led them to develop along different pathways to have eventually different jobs. And so you can see that happening here, that we're going to get slightly different signals from the pre-TCR that just has beta versus a gamma delta TCR that has a gamma and delta. And so that's going to push the cell to develop in a slightly different way. Yes, Shelby.
So the alpha chain acts as the pre-light chain or the surrogate light chain?
So it's called pre-T alpha that acts as a surrogate light chain.
So if it's an alpha beta?
If it's an alpha beta, we have not yet made the alpha chain. So it does both at the same time. Gamma deltas are weird. So you can see we're doing gamma delta. We're actually doing heavy and light at the same time, and gamma deltas, they're weird. Or we get beta, sort of a nice classic heavy chain. As I mentioned before, that cell that makes the beta, makes the heavy chain, still could, if it has to, switch to being a gamma delta. But most likely it's going to be an alpha beta. And the thing that finally makes that decision, the thing that finally pushes this cell to be totally an alpha beta and not have the chance to go back, is when we do that light chain rearrangement and when we make alpha. Because when we do our alpha chain rearrangements, remember that the entire delta locus is inside of the alpha locus. So when we put a V alpha and a J alpha together, we delete out the entire delta locus. And so once we delete delta, being a gamma delta is not a choice anymore. We have fully 100% committed. We can't go backwards and try something else. Now we are stuck being an alpha beta. As you can see in this image, these sort of choices are really happening at DN3, our DN3 cell. And you can also see this sort of here. Our DN3 and DN4s are the ones that are really starting to test that heavy chain. They're the ones that are really start doing the allelic exclusion and the proliferation, like right at as sort of we're transitioning. And then we get two other signals that are starting to happen in our pre-T cell, from our pre-T cell receptor, once we've made this decision. You can see these signals here. So we stopped beta chain rearrangement. We did allelic exclusion. This is sort of earlier in the DN3 time. We stimulated proliferation. Again, this is sort of a DN3 thing. We're going to survive. This is sort of a DN3 thing. We're going to stop making pre-T alpha because we don't need it anymore because we just used it. We're done using it. We can stop making it. And now we can actually start to rearrange our TCR alpha chain. When we start rearranging our TCR alpha chain, we also start transcribing and translating both CD4 and CD8 so that we can now do some signaling. And so as you can see, once we actually are fully doing that TCR alpha rearrangement, once we're kind of at the later stages of all these steps and we're really doing TCR alpha rearrangement, we're also making CD4 and CD8. And so now the cell is going to be a double positive cell because it makes both CD4 and CD8. And so we're now going to be at our double positive stage where we have both CD4 and CD8 made. And as you can see, when we have our double positives, we're now kind of moving across the cortex. We're moving basically towards the medulla. We've been doing a lot of these actions in the cortex. Now I want you to think for a second about a couple of stages that we've talked about. When I told you about those DN3 cells, DN3 to DN4 cells, one of the things I told you was happening in that DN3, DN4 pre-T cell is that the cell is going to proliferate. What happens to the area where this is happening when the cell proliferates? It's related to something I already said earlier. Yeah. It gets full of cells. Remember how we talked about how one part of the thing is full of a lot of cells, one part is not? We're going to suddenly get way more cells than we had before, right? Remember, Brenna, how you asked me about the double negatives? You asked me about DN3 and DN4. Where does proliferation happen? DN3 and DN4. And so so that's actually goes back to your question, and that's why there's so many more of those cells, because the cells actually start proliferating, and so we get way more there. That proliferation is sort of happening near the end of being a double negative, as the cells are going to be double positives. And so you can see there's way more double positives than double negatives. What you can also notice is that once we get to our double positive, we're going to get our full T cell receptor with an alpha chain and a beta chain on the surface of the cell. When we get and we are now totally 100% a double positive, what's the first thing that the cell is going to do when it makes a correct light chain? Test it. And if it finds out when it did its testing that it was correct, what do you probably want to do to that cell? You're going to actually now get some more proliferation and more survival. And so that's the other reason that we have so many double positives, is that we're going to get this massive proliferation and survival burst again. Because, hooray, we have successfully made a T cell receptor. Josh, I saw you had a question.
So are DN4 and double negative the same?
Double DN in DN4 is double negative. So basically what we're doing with DN1 through 4 is we're dividing up double negative into subtypes. Yeah, David.
Does the delta gamma T cells, do they have CD4 and CD8?
Rarely. We're just we'll see them later this semester. Otherwise, they've left the building at this point. So mostly one of the places we talk about them a lot is with where they're developing with that gamma or the delta versus alpha chain. We'll see them again later, but right now we're totally in alpha beta land at this point. Yeah, Brenna.
Brief reminder of
Yep.
what proliferation is. I think I'm confusing it with the signal.
Proliferation means cell division. It means cells making lots more copies of each other. And it's key that we don't have RAG on when we do proliferation, because we don't want to damage the DNA when we're also replicating the DNA. That would be kind of catastrophic. So you always have to have those two things separated. Yep, Shelby. Mm-hmm. Yeah, so the cortex is way full. The cortex is more dense than the medulla. And so if we look here, way back here, this is cortex, and this is medulla. Or that I can't reach is cortex, and then down here is medulla. Because it has more cells, because that's where they're proliferating. It proliferates after the beta chain and after the alpha chain. Like, you made a good beta chain, nice job. And now you made a good alpha chain, nice job. So now we have to deal with T cell central tolerance. So now when we are in the double positive stage, these cells we can think of as sort of immature T cells that are getting ready to do central tolerance, where we're basically what we want to do is we want to get rid of any T cell that would react to self, so that we don't get autoimmunity. So you saw the central tolerance process in B cells as well. And so we need to think about T cell central tolerance because it's fun and exciting. So when we think about T cells central tolerance and when we think about T cells testing, we actually have to have the T cell testing for a few things when it made its T cell receptor. As you're going to see when we talk about this, we're going to see that there are 4 options for what can happen to the T cell. One of them I'm just going to leave as like an option right now, and it will get filled in more next week. But one thing you could imagine is that the T cell receptor binds nothing in the thymus. If the T cell receptor can't bind to absolutely anything in the thymus, that means it can't get a signal from self MHC. Like it's not binding to your MHC. It can't. It like did VDJ recombination and made a receptor that binds to chalkboards and not MHC. Like it's so weird. It's so not MHC. So you could get a situation where it doesn't bind to MHC at all. Do you want that T cell that doesn't bind to MHC? No. So T cell binds nothing in the thymus. So we're going to call this absolutely no signal. Sometimes we talk about these T cells as useless. Oh, if you think that's funny, you're going to love some of the next stuff that's going to happen. So we're going to call this, we're going to think about the cell as being basically useless. Doesn't even bind to MHC. So we've got this. Now let's imagine that the T cell binds something really, 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 really strongly in the thymus, and it gets tons of signal. What's happening there? Go for it.
Is it reacting to self and there's so many in the thymus?
It's reacting really, really strongly to self. It's like, hello, I love you, self. Right? It's reacting really, really strongly to self. Getting tons of signal from that self. What would happen if this cell left the thymus and went out into the periphery, into the real world? Yeah, Shelby. Because it's going to bind and then it leaves and would potentially bind something else. So what would happen if this cell that binds real strong in the thymus leaves the thymus, gets to graduate from thymus college, and goes out into the real world? What would happen? I see some head shakes. Yeah, David.
It would respond to all your self cells.
It would respond, maybe not to all your self cells, but it would respond really strongly to some self antigen. It would give you autoimmunity, right? This cell is actively harmful. This cell is, like I said, actively harmful. It binds self a lot. You could end up with autoimmunity from this cell. So we actually don't want either of these scenarios in the thymus. One thing I'm going to tell you, there is a situation where we have the T cell binding sort of a medium high, because there are 4 outcomes here. That's something we're going to deal with next week. So I'm going to just tell you that medium high is a choice. And that's all I'm going to say about that choice right now. So we actually have to make sure that we are thinking about, we don't want you to bind to self really, really strongly in the thymus. That would be bad. But it would also be bad if you bound nothing in the thymus. So what we end up having happen in the thymus is that T cells are sort of looking at amount of signal. And the official name for this whole situation is called the Goldilocks model of T cell development. We're getting a situation where we're checking, are you self-reactive? If you're self-reactive, we got to kill you. We are actively going to kill you if you're self-reactive. But we also have to make sure you bind something and get some signal. And so what we actually do is that, and I've sort of told you this before, imagine these developing lymphocytes, and this is true of all developing lymphocytes, are super, super needy. They need some kind of pats on the head to tell them, you're good, you should survive. If a developing T cell cannot get any signal in the thymus, if it binds absolutely nothing in the thymus, it gets no signal. It is useless, and it undergoes a process that we refer to as death by neglect. Sometimes we also say that we ignore the useless cells. If a cell gets a low amount of signal, in the thymus, then that cell is useful. It binds to some stuff. And we're going to keep that cell. We're going to positively select that cell. So this cell has to get at least some signal in the thymus, has to get a light signal in the thymus so that it can be positively selected. And so sometimes we say select the useful or keep the useful. And this is based on the cell getting a light, low signal in the thymus. There's the high, that's a later problem. We also sometimes say that the cells that are undergoing death by neglect, sometimes we also refer to that as cells that fail positive selection, or a failure of positive selection. You got nothing. Alternatively, and so you can see a lot of cells are going to fail to be positively selected. They're going to die. Over 90% of developing T cells in the thymus die. I told you that this other book that I talked about calls the thymus murder university. That's part of what's going on. You also can guess that some of the later, if you remember the later parts of the thymus and the later subsets that we saw, have fewer cells. So the cells died. There are some that are going to actually become our T cells. It's like 2 to 5% of developing thymocytes actually get this low signal and become T cells. Some of them have this other thing. And then some of them bind really, 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 really strongly. That could mean that they bind to like everything, or they bind to one thing so strongly. In either case, they're getting a huge signal. And we are going to actively induce apoptosis. So we are going to negatively select these cells. Or sometimes we say kill the harmful. I have also heard people refer to death by neglect as dying of unrequited love or withering on the vine. And so you can see this happening here, the low, high, intermediate. It's not showing no binding in this figure. Yes, Jonah.
Just to make sure, this is happening in double positive.
This is happening in double positives. Yes. Yes, Josh.
So at this point, do you have like cells present the MHC in order to determine if the T cells are going to bind to their MHC?
So, great question. So there are a couple things we got to think about that are really related to Josh's question. So one of the things here is we've got, remember all those structural cells of the thymus that are all around the developing T cells? They're cells. They are nucleated cells. And so they all present on class 1, and they're just presenting stuff on class 1. And so that's how we're, making sure we can get a little bit of signal and recognize class 1. I also told you that when we talked about class 2, in terms of class 2 expression, I said there were the 3 professional antigen presenting cells, B cells, dendritic cells, and macrophages. And I said, oh yeah, also some cells in the thymus, specifically some cells in the thymus medulla. And so there are cells presenting on class 2 so that the cell can test on class 2. And so, all right, I'm going to do it this way. There's something I've said but haven't totally said here. I'm trying to decide which way I want to do this. All right, so again, one of the things we have to have is if we have weak or no binding, we have death by neglect. We have, the sort of lowish binding, then we actually positively select, give some signal to that T cell. That becomes a T cell that is able to eventually leave the thymus. This is how T cells learn what self-MHC is. Because the only MHC they have to test against in the thymus is your MHC. And so they are actually specifically tested to see, did you get a signal from the self-MHC? You will see a couple more slides like this later. But what I'm showing you here is a situation where I have mice of 3 different strains: orange, yellow, and blue. Here's my yellow mouse, here's my blue mouse. My yellow mouse undergoes radiation. When my yellow mouse undergoes radiation, that kills all of the rapidly dividing cells in the yellow mouse. That means the yellow mouse has no more immune system. Rapidly dividing. My blue mouse also gets radiation. So we have basically erased its immune system because we've gotten rid of all rapidly dividing cells. So if we just left these mice alone, they would die. And that would be very sad. We're going to save these mice, however. And we're going to save these mice by giving them a bone marrow transplant. When we do that, because I don't know, this is just all we had, we're going to use bone marrow from a different, genetically different mouse, an orange mouse. If I had left that bone marrow in the orange mouse, that bone marrow would go into the orange mouse's thymus, and that bone marrow would learn orange is self, because it's in the orange mouse's thymus. If, however, I take that orange bone marrow and I put it in the yellow mouse, radiation killed the immune system, but it didn't kill the structure of the thymus. So those orange cells are going to grow up in a yellow thymus, and they're going to say, yellow is self. Because they had, in order to get out of the thymus, in order to survive T-cell development and be positively selected, they had to interact with this thymus. It was yellow, so they now have learned that yellow is self. They are yellow restricted. That same orange bone marrow, if I put it into a blue mouse, it learns blue is self.
And it just forgets that one.
Yeah, it doesn't know what self is until it learns in the thymus.
Oh, so you're taking it before.
You're taking it before. You're taking the stem cells. And so I'm going to shoot to a couple of later slides that basically are showing the same thing. We can also do thymus transplantation. So there are even patients who don't have thymuses. It's a disease called DeGeorge syndrome, and we can actually just transplant new thymuses into them. And you don't even have to put it in the chest. You actually put it in the thigh, and it worked perfectly. There are mice that naturally don't have thymuses, which I can tell you more about later. But so if you have a mouse that doesn't have a thymus, and you can put in a thymus, if the thymus doesn't genetically match, like if you have a yellow mouse but you put in a green thymus, all those T cells are going to think green is self. If you have a green mouse and you give it yellow bone marrow, well, the thymus was green, so this mouse is going to always think that green, or this mouse is going to think that green, again, we're going to get green as self, even though these came from the yellow mouse. So whenever we do T cell development in the thymus, the resulting T cells always have the restriction of the thymus that they came from. So if we do weird genetics where the thymus has one kind of genetics and the bone marrow has another kind, the T cells always learn that whatever the thymus had is self. And that's because they had to be able to interact with the MHC of the thymus to get positively selected and leave the cell. Yes, Brian.
So how does thymus transplantation work?
Matching. When we talk about transplants, we'll get there. Basically, if in general we do match it pretty closely, but the reason why we have to do matching in a thymus transplant is different than the reason why we have to do matching in other transplants. Because in a thymus transplant, you're actually going to mess up the immune system. With, say, a kidney transplant, you're going to mess up the kidney.
Matching in the sense of matching the cell.
Matching the MHCs. Yeah. So there's basically, if your bone marrow transplantation also has its own unique rules. And so if you're transplanting immune stuff versus non-immune stuff, they're different rules. And you can also see that if our developing cell binds really tightly to self, then it's going to die because it's going to be killed as a harmful cell undergoing negative selection. These processes of selection, of negative and positive selection, are happening kind of at the end of the time that the cell is in the cortex, but very much when we're in the medulla. And so that's why the medulla is so much less dense than the cortex, because we are killing all the cells. And so, yes, you can see some of this is happening in the cortex, more of it's happening in the medulla. But now I have to break your brain a little bit. So, does this whole little scheme that I've written here make sense to you? about we bind nothing in the thymus and we bind super strongly in the thymus versus we get a little binding in the thymus? Yeah? What are the things that the T cell receptor, what is the T cell receptor binding in the thymus? What are like its options, its choices of stuff to bind in the thymus? What does it have on the menu to bind? The stroma cells. What do the stroma cells have in terms of like antigens? What was that? Self-antigens. There's not non-self stuff in the thymus. Actually, if there is non-self stuff in the thymus, it actually is a disaster. All that's binding in the thymus is to self. So we're testing these T cells. Do you bind to self 0? Do you bind to self a lot? Or do you bind to self a little? Every T cell that you have in your periphery had to get through positive selection in the thymus. That means every T cell in your entire body binds to self a little. It doesn't bind a lot. We killed those. But it has to bind a little to get positively selected in the thymus. Because that's the only choice you got in the thymus of like things for binding. People often do not like this. So what questions do you have, or why are you making cranky faces at me? Yes. If there was a pathogen in the thymus, you would probably negatively select all of the T cells in response to it, and so it would be terrible. But this is before that. This is just every T cell that leaves the thymus is a little bit self-reactive. Every single T cell that leaves the thymus is a little bit self-reactive. Why is this a thing that we have to think about? Why is this a potential problem? Yeah, Jonah.
Why don't we have autoimmunity?
Every T cell in your body is a little self-reactive. Every T cell in your body could lead to an autoimmune disease a little bit. We have this process that gives us the best, most broad set of T cells that happen to bind to MHC and work. We get rid of the most dangerous ones, but all the rest are a bit dangerous. And so we're basically balancing on the edge of autoimmunity. And peripheral tolerance becomes really important for these T cells because every single one is a little bit self-reactive. The idea is that they're sort of very, very weakly binding to self. And since there's only so much variation in biochemistry, there's only 20 amino acids, if you bind weakly to self, you might bind really strongly to a pathogen that's sort of similar but not exactly the same. So the hope is that if we keep all these, they bound MHC, hopefully they bind some kind of pathogen that we've never seen before. We got rid of the ones that are going to be really, really, really dangerous, or we did something else with the ones that are going to be really, really, really dangerous. But every single T cell in our body predisposed is a little bit self-reactive, and so we are automatically, because of how T cell development worked, predisposed to autoimmunity. And so, if we live long enough, we're having more and more chances for peripheral tolerance to get messed up, and for that T cell to now cause a problem where it didn't before. We've got one other detail about antigens in the thymus that we need to talk about, which I'll tell you more about on Monday. And we'll finish a few details about T cell development. And then we will finally be in the periphery with all of our cells, and we will be able to actually start thinking about how we make a response to a pathogen. I'll see you guys on Monday, and please remember your problem set by 5.